Hello, good evening. My name is Diana Munn. I'm Director of Public Programs at the Harvard Museums of Science and Culture. It is my pleasure to welcome you to tonight's Evolution Matters lecture, sponsored by the Harvard Museum of Natural History. We are delighted to have Professor Javier Ortega-Hernández with us tonight, who will discuss some of the earliest known organisms from the Cambrian period and their role in the evolution of animals. I would like to take a moment to recognize Drs. Herman and Joan Suit for their generous sponsorship of this lecture series. They have sponsored the Evolution Matters lecture series for the past 10 years, and it is their support that allows us to present and record these lectures. It is now my pleasure to introduce James Henkin, Professor of Biology, Curator of Herpetology, Alexander Agassiz Professor of Zoology, and Director of the Museum of Comparative Zoology at Harvard University, who will introduce our speaker.
Thank you, Diana. I want to welcome all of you as well to joining us this evening. I look forward to the lecture as much as you do. Now, before I introduce our speaker, I've been asked to introduce my background, and then we'll move on from there. I'm speaking to you from Arlington, Virginia, and as many of you may have read, one of the unexpected consequences or side effects of the COVID-19 pandemic following the lack of activity in our cities is that a lot of wildlife returned to places where they hadn't been very conspicuous in a long time. And that was the case in our house here in Arlington, where a pair of black vultures set up shop in the attic of our garage in our home in April. And what were four birds eventually became, what started out as two birds about a month ago became four birds. And you see those four birds in the photo behind me. That's a picture I took just yesterday on our back porch. They've become quite fond of us, especially once we started feeding them raw chicken for breakfast every morning in the backyard. Anyway, that's my social life, or our social life, for the last few months, and I hope you don't mind looking at them. Now, on to our speaker for this evening. Javier Ortega-Hernández is a invertebrate paleontologist who is interested in the evolution of major groups of animals, not living animals so much like the vultures you see behind me, but animals that evolved right at the dawn of animal evolution. Now, to do this, to study organisms like this, you have to work typically in what's called the Paleozoic era, and especially focus on events such as the Cambrian explosion and the so-called Great Ordovician biodiversification event, which I suspect Javier will be talking to us about a lot this evening. Now, Javier is originally from Mexico City, where he received an undergraduate degree in biology from the Universidad Nacional Autónoma de Mexico. In 2008, he moved across the big pond over to the United Kingdom, where he completed a master's degree in paleobiology at the University of Bristol. He then stayed in England. He moved over to the University of Cambridge, where he completed a PhD in earth sciences in 2013, but then stayed on another 5 years when he was offered a very prestigious postdoctoral research fellowship called the Herschel Smith Postdoctoral Research Fellowship in Biological Sciences, also at Cambridge. He joined Harvard in 2019 as an assistant professor in the Department of Organismic and Evolutionary Biology, and at the same time, a curator, or the curator, of invertebrate paleontology in the Museum of Comparative Zoology, where I'm director. And it's been a pleasure to have Javier with us these last couple of years, together with Stephanie Pierce, our curator of vertebrate paleontology, who joined the faculty in the museum only a few years before Javier did. The two of them have invigorated the study of paleobiology at Harvard, and they've both been a wonderful addition to MCZ. Now, as you'll see, I suspect you'll see from tonight's lecture, Javier is quite a well-rounded scientist. He combines the study of fossils, newly collected fossils that he gets himself during field work, with laboratory investigations that use some very modern techniques to see things in fossils, quite literally, that people hadn't been able to see until a few years ago. So without any further ado, let's hear what Javier has to say. Javier?
Thank you so much, Jim. That was a lovely introduction. I appreciate it. And thank you so much, Diana. It is a real pleasure to be here. And I am super happy to be able to talk to all of you today. This is going to be, I hope, a fun lecture. And can I just check that everything looks okay on everyone's side? Cool, okay. So let's get started. So today I'm going to talk about wonderful Cambrian beasts. But before I do that, I want to wish all of you a happy National Fossil Day. Like many of you may know, there is a National Fossil Day in the United States, and it is celebrated on the second Wednesday of October. And this particular year, the artwork for National Fossil Day is these Permian reefs at Guadalupe and Glass Mountains in Texas and New Mexico. So it is very special for me to be able to show this particularly paleontological day with all of you, and I hope that you will enjoy the lecture going forward. So, as Jim mentioned, I am an invertebrate paleobiologist, and I specialized on the Cambrian explosion and all the events that surround it, and particularly all the animals associated with it. As many people interested in the Cambrian explosion, I came to this line of work through reading Wonderful Life by Steve J. Gould, who was the curator of invertebrate paleontology a few years before myself, also here in Harvard. And here we have a picture of him in 1987. Now, Steve Gould became very influential, even though he didn't actually work directly on the Cambrian itself, because he did this very in-depth exploration of what all of these studies being produced around the 70s and 80s regarding the Cambrian explosion animals from the Brugge shell and what they meant. And him being kind of famous for his writing, he did combine a lot of hyperbole and a little bit of pop culture in trying to make some of his writings more accessible, but also to try to make a point about the importance of these organisms from an evolutionary point of view. And this is just something I want to show, one of my favorite paragraphs from Wonderful Life, which is where he sort of emphasizes that many of these animals in the Cambrian are what we now know very sort of informally as weird wonders. So in this particular case, he's talking about what defines the success of some groups of animals over others. And specifically, he argues, well, if we extend this theme, being, again, the difficulties in regarding what makes animals be successful or not, beyond the arthropods, the weird wonders of the Brugge shell, we want to ask ourselves, why not Topovinia and Wiwaxia, our survivors? Why don't we have a world of grazing marine herbivores with sclerates and not snail shells? Why not Anomalocaris, a very famous Cambrian animal, and a world of marine predators with grasping limbs with a jaw like nutcracker? And he goes on to say, why didn't Steven Spielberg, why don't we have now a Steven Spielberg movie that features an Anomalocaris that would chomp on a crusty kind of sailor instead of having a shark? And this is just a wonderful sort of exploration of what could have been if history would have taken another route. However, what is interesting here is that Gould gets a lot of recognition for writing Wonderful Life. However, it was actually a fundamentally collaborative effort because, of course, Gould provided the narrative, but one of the reasons Wonderful Life became so influential was because it really did bring to life many of these Cambrian weird animals for the first time. And that was thanks to the wonderful illustrations by the illustrator Marianne Collins. So a lot of the figures I'm going to show you right now are Marianne's work. And it was really instrumental because it helped to bridge this gap between what is really a very technical scientific illustration with a much more lifelike organism that we can much easily relate to. And this is the only time I have met Marianne. This was in 2009, and we're standing in the Walcott Quarry in British Columbia with Pika Mountain just at my back. So this was a very special moment for me a few years ago. And it's something that, again, I want to emphasize that Marianne did have a truly impactful contribution to the way that we see the Cambrian explosion right now. So as I mentioned before, these are a whole bunch of Cambrian weird wonders, as we would very informally call them. And this includes forms that are really difficult, traditionally speaking, to link with extant diversity nowadays. They are weird and have strange shapes and peculiar ecologies, and they're just kind of difficult to figure out. And again, because we have these wonderful illustrations, we have a much better sense of, well, what these look like as animals. So now we jump in time, and now we can have a lot more research, a lot more technology informing what life was like in the Cambrian. And this is just a wonderful example of a 3D reconstruction produced by our colleagues at the Royal Ontario Museum that shows what the Bruges shell environment would have looked like. Again, this marine ecosystem full of strange animals that we don't see around anymore, and that are nevertheless still sparking our imagination. Of course, we have Anomalocaris doing a victory lap after eating a trilobite. So this is what we think about when we think about the Cambrian explosion nowadays. However, all of this is possible because of the availability of diverse and very abundant fossils of non-shelly complex animals in what we call exceptional deposits. These are sites like the Bruges shell and a few more I'm going to mention in this talk. And what is really remarkable here is that all of these organisms are fundamentally soft and fundamentally prone to decay under normal circumstances. In most of the fossil record, we will see shells and bones and teeth, but only under the most special circumstances we will actually get a lot of this diversity. So it makes it that the study of these deposits is actually very important because it allows us to have this unique window into life that we would otherwise never would be able to see and appreciate. And the best part is that we can actually find this sort of deposits all around the world. Of course, one of the more famous ones is the Bruges shell, which is Middle Cambrian in British Columbia in the Canadian Rockies. And again, we have Walcott Quarry on the left with Pica Mountain on the back. And Bruges shell fossils are pretty flat. I mean, they're super pancaked. And that makes it so that they have some difficulties in studying them, but nevertheless, they still remain as some of the best preserved fossils that anyone can ever see because of the amount of morphological detail that they have. However, also we have a whole bunch of other localities around the world. One that myself and several people in my research group are very invested in working on, including Rudy Leroy-Abril, Stephen Pates, are fossils from the Western USA, particularly in the area we call the House Range, which hosts at least half a dozen of exceptional fossiliferous deposits. This may not have the beautiful, amazing detailed preservation of the Bruges shell, but they are nevertheless providing a much greater geographical over over which we can find these fossils. And we find very different organisms that we don't really see in the Bruges shell. So it really complements our view of Cambrian life much more effectively. And of course, that doesn't stay in North America. We can also go to North Greenland with the Sirius Passage. We can go to South Australia with the Emu Bay shell. We can go to several parts of China, in this particular case with the Qingyang biota. And we have, again, this very consistent record during the Cambrian in that we have some sites which are just truly exceptional, and they preserve all of these weird wonders, these strange animals, sometimes in great numbers as well. So they are an amazing, invaluable resource for us to really understand how the early biosphere came to be, and basically how complex life started, which again, I think has a fundamental intrinsic value for us to know where everything that is surrounding us looks like. Where does it come from, and why the biosphere looks the way it does. Now, one of the great complications of working with the Cambrian explosion and this kind of organisms, and something that Gould became sort of infamous in his book Wonderful Life, is again emphasizing the difficulties of linking directly in an evolutionary relationship all of these weird oddballs from the Cambrian with the biodiversity that we see today. Because we are living in the present, of course, we have a much better understanding of what biodiversity looks like, works like, feels like, and smells like nowadays. So we can do all sorts of fancy studies on them, including molecular biology, and you can dissect the whole thing. With the Cambrian, that is not so much possible. We only have the morphology and maybe a little bit of chemical information. So it really is difficult to be able to say, well, are all of these Cambrian oddballs a whole different thing? Are they this so-called failed evolutionary experiment? Are they just different kingdoms or phyla that didn't go anywhere? Or are they something else? And one of the things I do quite often, and a lot of the people in our research group do, is that we strive to find the connections that allow us to understand Cambrian forms in the context of the extant biosphere. So with that in mind, I'm going to show you a few case studies, which are a combination of an old classic from the Burgess Shell with a little bit of a twist. And on the second half of the talk, I will show some of the more current research that we are doing to understand what these Cambrian organisms are and how they can inform the evolution of life on Earth. So this is important because we know that all of the major groups of animals, or most of the major groups of animals, started in the Cambrian, or at least have a record in the Cambrian. So being able to tie, again, all of this extant diversity with all of these wacky fossils is super useful, because then one can inform the other one. So again, this is just part of the reason why we spend a lot of our time and lose some nights of sleep thinking about flattened fossils that may look really great in the centerpiece, but they're actually much more important than that. And they do provide this, again, invaluable source of information. So with that being said, the first thing I want to talk about is this quirky story of Hallucigenia sparsa. Now, Hallucigenia is a classic. This has been going on for a few years now, and it is probably one of the most famous animals from the Cambrian. So, and the reason before that is because it has this kind of really fun history behind it. So Hallucigenia was originally found by Charles Lowell Walcott at the start of the last century, and it was initially sort of classified as some sort of worm. When it was revised in the 70s by colleagues at Cambridge, particularly by Simon Conway Morris, it was interpreted in this particular orientation. And well, as you can probably tell, it's pretty difficult to say what it is. It's kind of all over the place. It has some spikes, it has what appears to be some tentacles, it looks a bit like a sausage in the middle. It's not super obvious what it is. So based on this initial description, Simon produced this, again, now quite notorious reconstruction and suggested this name Hallucigenia because this looks like a hallucination. So this is one of those organisms that Gould really drove home, that it's so different and so surreal that it just cannot have any links with anything living anymore, or anything living today. However, when we look a little bit closer, we start to find some characters that actually allow us to link it with some groups that make a little bit more sense. And just before we go further, I always want to emphasize that in the 70s, there was absolutely no context for interpreting many of these fossils. So it is not it's usually fun to say like, oh, well, how silly, people got it wrong. But again, in the 70s, there was nothing to compare it to. So any interpretation was as good as the next one. Some years pass, and then in 1992, someone had in good measure, and not someone, Lars Ramskjöld, had in good measure to actually take a dentist drill and actually prepare some of the fossil to try to reveal some of the features hidden within the rock itself. And what he did is that he started to follow some of these tentacle-like things on the lower half of the animal, and he discovered claws. This became quite useful because during the 80s and early 90s, a lot of fossils from the Chengjiang biota in China were discovered, and there was this group of animals called the Lobopodians, which are basically worms with legs, that started to be found. So this allowed people to realize, or it allowed Lars Ramskjöld to realize, like, well, Hallucigenia is not some sort of weird thing, it's actually a Lobopodian. And it was possible to make a correct side-up reconstruction, where now we interpret this animal as having this dorsal, or these armor spines on the back, having these pairs of legs with claws at the underside, having a more or less featureless head, and then doing its own thing. It's still pretty weird. And if we were going to stop it at that, it would still be difficult to argue, well, this is clearly related to anything living around today. So a few years ago, a colleague of mine, Martin Smith, who is a professor at Durham University in the UK, we decided to take another look at Hallucigenia, and specifically to use some new techniques to interpret its morphology, or to analyze its morphology. So one of the features of Hallucigenia, now illustrated on the right way up, is that it has these very prominent spines, as I mentioned before, and it also has this very prominent claws. And you may be able to see these small hook-like projections, which are very, very dark on the underside. And they look very dark because they are preserved as carbon films. So the carbon looks, it's dark, it has been graphitized because of the way that the brookish shell is formed. And because it's made of carbon, it is actually possible to analyze it under an electron microscope to see some of these details. And what you have here is a really magnified single claw of Hallucigenia. What becomes very obvious is that in addition to having this very distinctive sickle shape, it also has a number of features inside of it. So we were able to identify up to 3 constituent elements, which in this case you can think about of stacked ice cream cones, just one inside of the other one. So this was quite fun, but also really remarkable because after doing quite a bit of research on the literature, we only found one group of animals nowadays that has claws, that looks like a worm, and has this overall constitution, and that also has claws with this particular organization. And the answer is the velvet worms, or onychophorans. Velvet worms, some of you may know because they are extremely cute, as you can probably appreciate. And they are very small, well, they're reasonably small invertebrates. They are restricted to tropical environments. They are exclusively terrestrial. And even though they look a bit like caterpillars, they are not insects whatsoever. They are their own thing. They have a very ancient history. And they're just kind of really fun little animals. They spit slime, Spider-Man style, using papillae on their mouth to capture prey. And what was, again, quite significant is that we were able to find the link that connects Hallucigenia with onychophorans. So if we see the claws in the legs of onychophorans and the jaws inside their mouth, they have this very similar structure that we see in Hallucigenia. So on the left side, we have this lovely 3D image of an onychophoran leg, which has 2 claws on the tip. And then on the bottom, we have a small video of an onychophoran kind of showing its jaw on the mouth. And what you can see on the right is just a dissected version of that. And again, we have this very distinctive sort of claw-in-claw or cone-in-cone construction, both in the claws and in the jaws. The jaws in this case are a modified set of legs, so it does make sense that it has the same organization. So because we can find this complex feature in both of these animals, we can actually tie Hallucigenia with the onychophorans. And by default, we can say something about the early history of onychophorans themselves, which otherwise have a very meager fossil record. They only are known from very few amber deposits and only one decent carboniferous macrofossil, but it still does not have a lot of detail. So this is telling us that at least some relatives of onychophorans, even though they are very distant relatives, do extend to the Cambrian explosion. And this is implying that onychophorans throughout their history had to lose these offensive spines and had to evolve these internal jaws for feeding, maybe associated with the terrestrial environment. Again, this is really strange because Hallucigenia is marine and onychophorans are the only major group of animals that has no aquatic representatives nowadays. What was really fun about this is that, well, I mean, that is great for Hallucigenia, but Hallucigenia actually has a lot of relatives in the Cambrian, and some of the weirdest animals that you will ever see. And well, when we published this a few years ago, journalists when we called it Hallucigenia on steroids, because it sort of is. It's much bigger, it's much spikier and leggier and hairier and everything. So in this case, we have this animal, which is called Collincium ciliosum. And this is from the early Cambrian Xiaoxiba biota in South China. So this is an organism that we worked with our colleagues in Yunnan University a few years ago. And what you will notice is that, well, it is also a Lobopodian. However, it does have some other features. It has this very strong dorsal armature of spines, which are super big, and it has way more than Hallucigenia. But very prominently, the legs on the front have this almost tentacular and feathery-like appearance. They have all of these setae on the sides that make it look like a feather, which is super weird. This is a reconstruction of this animal, and I swear that every single thing you see in this reconstruction, you can see on the fossils. This has no artistic license whatsoever. Every single hair and leg and claw and spine you can see in this fossil. So as you can appreciate, this is, again, like Hallucigenia, but a little bit hyper. It has much more spines. It has these very peculiar limbs. And it is very interesting to think why. It is also interesting that this is not the only one. There's a lot of them. This is another representative, an animal called Asinocricus stichus from the Spence Shale in the USA. And this animal was originally interpreted as a green alga. But then after the discovery of some of these other Lobopodians, now we know it is also one of these very peculiar Woollyshanids. And this one is particularly metal because it has rings of spikes all the way through. This is by far the thorniest worm that you will find in the Cambrian. More recently, we have some research by our colleagues in the UK, by Richie Howard and Xiaoyama. And this is a redescription of this organism called Fascivermis junanicus, also from the Shenyang. Now, Fascivermis is also related to these animals, and we can tell it because it has this worm-like body and these very distinctive feathery limbs. But you will notice it has no spines, unlike Asinocricus and unlike Collencium or Hallucigenia. It is one of these suspension-feeding worms that has become naked and has lost the legs on its rear side. It only has this sort of pear-shaped bulb. So it is thought that this animal was actually living sort of like buried in the sediment and then just kind of did this movement with its arms to filter feed. And then we have yet another one, also from the Burgess Shale, of Ativermis cribatus, also described by our colleagues in the Royal Ontario Museum in Toronto. And again, it is similar to all of these animals as well. It has no spines. It does have claws on its bum, but it also has these kind of weird feathery tentacles. And our colleagues were so kind to produce this wonderful animated reconstruction that illustrates how it is thought that all of these animals fed. These animals are collectively known as Woollyshanids, which is the clade that we're talking about. And the idea is that they would use all of these feathery limbs to sort of like hug the water and basically bring together either small animals or a little bit of organic matter into their mouths. And you may be thinking, yeah, right. I mean, this looks like a complete stretch. But what is really cool is that this actually happens still today, and you may not appreciate in which animals it does happen. So here we have a video of a hermit crab, but the hermit crab has a cirripede or a barnacle associated with it, just kind of like attached, which is this kind of like cone-like thing that has these feathery plumose things sticking out. Now, barnacles are crustaceans, which are very heavily modified, and they basically swim around, stick themselves through their head to something, and then form a shell and start to filter feed. So what we have here is an arthropod, which is sort of closely related to Lobopodians, doing exactly the same thing that we think these weird Cambrian animals are doing. What happens here is that this is just an arthropod. Lobopodians are a different part of the tree of life, but we see fundamentally the same strategy. So it is not so strange anymore to think about some of these legged worms doing basically the same thing, just in a slightly, probably in a slower pace, because again, barnacles are quite tiny, so they can do these movements much quicker. So what this is telling us is that these Woollyshanids are reflecting a great diversity. They are extremely specialized suspension-feeding worm-like marine animals, and they have this global distribution. So they are just a wonderfully weird group that is doing its own thing back in the Cambrian. And nevertheless, again, they share so many characters with Hallucigenia, so we have to think, well, clearly these animals are also related to Hallucigenia and are also related by default with onychophorans. And what really kind of like springs to mind is that, well, what happened? Why did onychophorans sort of lose all of these accoutrements? I mean, they were so cool back in the Cambrian, and now they're cute, but they're definitely less versatile than they once were. And what you see on the left is a pretty good representation of extant onychophorans. And as you can appreciate, they are very colorful and they're, again, adorable, but they are very samey. They are a very homogeneous group of animals. So they did lose a lot of this complex ecology that they had back in the Cambrian. So if we put this into more quantitative terms, we can say that onychophorans basically became much less variable throughout their evolution compared to what happens in the Cambrian. Again, this graph is showing that Cambrian representatives occupied much greater degree of morphospace or ecological diversification compared to the representatives that we see nowadays. So the take-home message from this part of the talk is that Hallucigenia and its skin do reveal this rich and complex evolutionary history for onychophorans during the Cambrian explosion. And if it were not because of these exceptional fossils, we would have no idea that this happened. Because again, onychophorans are really soft and squishy, mushy animals. If we had some onychophorans in the lab, we didn't look after them super well, and they turned into snot. They disappeared. They were not even carcasses left behind. So that gives us a good idea of how difficult it is for these animals to be represented in the rock record. And it emphasizes, again, the importance of being able to have access to these particular deposits. Now, a quick kind of midway intermission. The next thing I'm going to talk about deals more with our current ongoing research, and this is a bit interactive. So what I want you to do is to grab your phone. I'm going to assume that most of you will have a phone accessible to you, and you will see in the next few slides a number of QR codes, like this circle that you see in your lower left corner. Now, if you use your phone, you open your camera app, and you point it towards the screen, your camera will recognize this QR code as it does a face, more or less. When prompted, you will be asked to open a link that is going to lead you to a website called Sketchfab. And you will be able to actually see and interact with the models I'm going to show you in this talk directly at the tip of your fingers, a little bit similar to this representation in the lower right. So again, I invite you all to do this. It's quite fun. I will still show the figures in the talk, and all of these links will be accessible for people after the talk as well, in case you don't have a phone next to you. With all of that being said, let's get started. So one of the big complications of working with Cambrian organisms is that, again, they're super flat for the most part. And we have to think about it a little bit hard, because at the end of the day, these are small invertebrate animals. They have become buried by thousands of tons of mud and sediment when they were buried. And then this sediment has become lithified, has become rock. Then it has gone into the earth crust a little bit and has experienced all of these intense tectonic and geological pressures. So it is understandable that these fossils are less than pristine. So it would be great to be able to maybe see a little bit more than meets the eye. And this is what we have strived to do with some new methods. So what I'm showing you here is a flyby across a fossil from the Cambrian, from the early Cambrian Chengjiang biota. So what we have been developing in close collaboration with our colleagues in Yunnan University in South China is the use of computed tomography to be able to understand the 3D morphology of these very flattened but not completely 2D fossils for the first time, and to be able to basically observe what is going on the side inside of the rock, like the dark side of the moon, if you may. This part that we would never be able to see with our eyes alone, but that we can get to using X-ray technology. We can do this because the fossils themselves are enriched in iron. The iron is a different density compared to the rock itself. It's a little bit similar to the way that we see ourselves in X-rays. Our bones are much denser than our flesh. So we can see, for example, right about now, you will start to see the fossil as this very bright white spot at the top of the image. So that is what we look for, and we're then able to reconstruct into a 3D model that allows us to understand the morphology of these animals in amazing detail. So let me show you some cool fossils again. In this particular case, we have been working with a few arthropods as of lately. And this has been useful for a number of reasons. One of them is that this technique allows us to restudy animals that were very poorly known, that either are extremely rare, or animals that are super well known, and that we still can find some new features to say, we can find some new features in them, and that is going to give us, again, that little extra information to understand them a little bit better. The first one I want to talk about is this organism called Arcucunia multinodosa, which was described in 1999, so a little bit over 20 years ago. And as you can appreciate from this drawing, this is as it was originally described. Granted, it is not the most detailed drawing, and it was a different time 20 years ago, but there it is. So again, you can use a QR code to scan this fossil and check it out in your mobile device. And again, just to emphasize the point, Arcucunia is a reasonably rare animal. It is not the rarest, but it is not super abundant, which is part of the reason it was sort of ignored for a long time. However, in the last few years, we were able to start this collaboration with our colleagues in China. A colleague of mine, Joanna Wolf, and myself went to South China, to Kunming, to Union University, to do this collaboration. And we were able to essentially, again, get an amazing glimpse into the hidden morphology of this animal that would not be possible based on its fossil alone. Again, on the left side, we have how the fossil looks, which is okay. I mean, it's not the best, but it's not horrible. But then on the right side, what we have is this tomographic virtual model. This is what we get when we actually process the data and we are able to study the other side of the material. And this is what you will be seeing in the tomographic model in Sketchfab as well. If we take a closer look, Arcucunia is full to the brim with lovely morphology, amazing detail, and really informative features. So we were able to identify that the antenna have this very sort of complex flattened CT, which look a little bit similar to some features we see in crustaceans nowadays, which are used for sensing both by touching and chemical sensation. We were able to find a number of additional limbs on the head. So for example, this feature in green is actually a second pair of antenna. We were able to see mandibles and other mouth parts. And we were also able to see the trunk limbs in really exceptional detail. And the trunk limbs is where Arcucunia absolutely shines, because it's possible to actually dissect them out individually because all of this is done virtually. And one of the things that you will notice is that it is flat, but it is still pretty three-dimensional. So what you're seeing on the left side is a model of the middle of the body, is a trunk. And this has not been modified by us. This is as flat as the animal is. Now, it is a tiny specimen. So for our size, it is essentially two-dimensional. But when we look close enough, you can see that there is a little bit of three-dimensionality in it. And that is quite valuable because it allows us to have a much better idea of the spatial organization of all of these different features. Now, on the right side, what we have is one dissected biremius limb of Arcucunia. And this is where it becomes a really interesting story, because we can identify the components of the limb in great detail. We have the green part, which is basically what we call the endopod, or the walking leg. We have the blue bit, which is the exopod, which has, it's a bit like a gill or like a respiratory structure. And then we found another feature that we don't usually see in many of these organisms, and that is this feature called the epipodite. Now, the epipodite looks a bit like a teardrop-shaped lobe, and it may not look like much, but it is actually super significant, because epipodites are features that have a respiratory and osmoregulatory function, and as far as we know from extant arthropods, they are only ever found in crustaceans. In this case, we have some lovely electron microscopy images of branchiopod larvae or juveniles that are showing the limbs developing. And what you can see, again, is this kind of leafy teardrop-shaped lobes basically at the shoulder of the limb, if you may. Like if my arm was a limb, they're just kind of like sticking out over here. And again, this is something we see in several groups of crustaceans nowadays, and we don't see them in any other group. We do not see them in insects, we do not see them in myriapods, we do not see them in chelicerates. So being able to find this complex feature, again, is telling us something significant about Arcucunia. And in particular, this is super important because it is going to produce this direct link between Arcucunia and crustaceans nowadays. There is a lot of people doing kind of really interesting work on molecular clock analysis and phylogeny, trying to understand what is the precise timing for the origin of all of these groups of animals. And many studies suggest that crustaceans did originate in the Cambrian. This particular case, this is true for decapods, but again, there is some uncertainty there, and they may have, at least in Ordovician, maybe Cambrian origin. But decapods are one of the groups of crustaceans that do have epipodites. So again, being able to find this feature in Arcucunia is really giving us this very strong signal that not only we have the epipodids, we have the head structure and the trunk appendages. So all of these features come together to tell us Arcucunia is very likely related to crustaceans, and this, again, comes in nice agreement with these molecular clock estimates. And it is always wonderful to have agreement between different sources of data, be it molecular, morphological, or developmental. When we have all the data telling the same story, this is very powerful because it does help strengthen the argument a lot more. So it just makes for a much more robust hypothesis. The second animal I want to tell you about today with these 3D methods is this organism called Cambrian. And you might think, oh, Cambrian is this shrimpy-looking thing, of which there are 2 in this lab. But you would be wrong. Cambrian is actually that little smirk of orange on the lower left side that looks not like much. But I saw it's actually super interesting. And this is, again, a really useful demonstration of how micro-CT can bring up some details in fossils which are, again, poorly preserved or super rare. And you cannot get more rare than Cambrian, which is known from this only specimen. So this is what Cambrian looks like. And admittedly, micro-CT didn't actually reveal the amazing details on the underside that we were hoping for, but it did help us to have a much better view of what the outline of this animal looks like and to have a much better idea of the original convexity. As you can tell, both from the light photograph and from the micro-CT model, there are a lot of compaction wrinkles associated with this. And this happens when we have a very convex structure. Think about like a bowl or a helmet, and this gets flat. And with that flattening, we have lines of weakness that are going to just kind of, because of the compression, they're going to be expressed in the fossil. So this is telling us that this animal in life was quite voluminous in its shape. Now, even though we do not have a lot of limb information, we only have this one piece of exoskeleton, we know for sure it is Cambrian because there was some really amazing research published last year which actually described the first Cambrian species, Cambrian falcatus from the Brugge shell. And some of you who might subscribe to Science may remember this journal cover from a few years ago. So what we have on the lower right is this lovely 3D model of Cambrian from the Brugge shell. And this shows how we understand its morphology. So the fossil I showed you before is basically the head shield of this animal that has this very peculiar shape that is hosting the animal underneath. And the animal itself is what we would call a radioland. This is an animal related to Anomalocaris, which is, again, very famous and well known. But it is a little bit different. It has these frontal appendages, which in this case have these kind of long spines, and we will discuss in a bit more detail. And what is also really fun is that, again, similar to the Lobopodians, there are several instances of Cambrian since its discovery last year. So on the upper right, we have Jean-Bernard Caron, the curator of Invertebrate Paleontology at Rome, holding this very large fossil that is, as my understanding is, affectionately known as the mothership, because it sort of looks a bit like a spaceship. And that is another relative of Cambrian. It may be a different species or a different genus altogether, but the shape of this head is unmistakable. So we are able to make this link directly because the preservation of this organism and the use of computer tomography quite effectively. Now, what is really interesting is when we put Cambrian into a larger context. As I mentioned before, Cambrian was first known from the Brugge shell, which is this red star in the middle Cambrian, which is approximately 580 million years old. And our new Cambrian is from the early Cambrian of China, the Chengjiang, which is approximately 10 million years older, so 518. Now, one of the things that they share is having this very distinctive horseshoe-shaped outline. And they are not alone in this distinction. Horseshoe-shaped outlines are also known in trilobites, like the one in the upper right, in this Harpeti trilobite. But also, it is the namesake of this animal in the lower left, in panel C, which is the horseshoe crab, which is neither a horseshoe nor a crab. It's actually a chelicerate. It is more related to arachnids. But the point here being is that we have these animals that are belonging to very different parts of the tree of life, And all of them have this very peculiar horseshoe-shaped head. And we have to ask ourselves, well, why are they doing that? Why is it so advantageous to have a horseshoe-shaped head? And this comes to demonstrate one of the more interesting aspects of radiodont evolution in the Cambrian, in that they are amazingly ecologically diverse. So radiodonts come in all sorts of sizes. They go from the Cambrian size, which is super tiny. The fossil is just like around a centimeter long. And we go all the way to the size of Aegirocassis, which is actually an Ordovician radiodont from Morocco, which can be essentially 2 meters long, like the actual specimens, and they are very difficult to carry in the field, of course, and everything in between. But then there is not only the size differential, there is also the ecology difference. And basically radiodonts come in 3 main flavors. They can either be filter feeders, in that they have this very elongate body, and they have these frontal limbs that they use for, again, swimming in the water column and eating, well, basically filtering the water as it goes. We have raptorial predators like Anomalocaris, which is, again, the more famous one, and they have more of the stabby frontal appendages with spines, so they would just kind of go and eat animals, either soft-bodied or maybe a bit lightly shelled. And then we have the sediment sifters, as it is the case of Cambrian. And these sediment sifters would actually swim very close to the bottom of the seafloor, and they would use their limbs to basically rake up the sediment and capture any animals or, again, small organisms living there and then consume them. So this is quite interesting, because what I'm essentially trying to explain is that back in the Cambrian, we have this group of arthropod relatives, which are essentially behaving on one end of the spectrum like basking sharks or baleen whales, in that they are quite large animals swimming in the water column, filling their mouth with filtered water to get all the tiny animals out. But then on the same group of Cambrian animals, we also have the whole other end of the spectrum. We have this very distinctive horseshoe-shaped organisms that are living very close to the bottom of the ocean or at the bottom of the ocean, and just basically, again, sweeping the Cambrian seafloors looking for food. And all of these belong to the same group. So we have this, again, amazing ecological diversity within a single group of organisms. So the take-home message here is that even though we only know this animal Cambrian in China from a single specimen, and a single not great specimen, we can still tell a lot about how this group diversified and how they lived. And it demonstrates that very early on during the evolution of radiodonts, they had already basically explored either most or all of the ecological options available to them that we will see much later in the Cambrian as well, which is, again, quite exciting. Now, the last thing I will talk about rather briefly is this animal called Liancolia sabrosa. And what I'd like to say at this point of the talk is that if there is just one QR code that you want to scan, is this one, because this animal came out absolutely beautifully on the scan. So it is really worth your time if, again, you are into this sort of thing. Now, one of the things with Liancolia sabrosa, also from the Chengjiang, is that it is actually very well known. Liancolia has been, this particular taxon is known from hundreds of specimens. There are at least half a dozen publications that deal with its morphology, ontogeny, ecology. So it is really one of those animals that we would think, well, why even bother? We know everything there is to know about this organism. So let's put it in the drawer, let's move on to the next one. But again, one of the beauties of using this approach of micro-CT to Chengjiang fossils is that it still can provide some surprises, even in specimens which are in species that are so well known. And in this particular case, one of the standing controversies with Liancolia and other animals related to it called Megacheirans is, where do they go in the tree of life? Now, what we see on the left is yet another of these great videos by the ROM, in the ROM website, showing Liancolia superlata. It is a close relative from the Brugge shell. but it basically has the same bits and bobs. Is this animal? It looks a bit like a woodlouse, but it lives in the oceans. And they have these very distinctive, very, very big, what we call great appendages, that in this case look like weepy things on the front. Now, the origin of these great appendages has been super debated and really, really, really controversial. And the reason for that is that depending on where they go, or depending on the interpretations of where these animals go in the tree of life, we have to make entirely different reconstructions of the evolution of arthropods. So we have a vested interest in kind of straightening the record and being able to say where actually these animals go. And the two main interpretations are, well, either they are related to arthropods, but distantly, or they are maybe closer relatives to chelicerates. And all of that basically depends on how we interpret the origin in the head of this first pair of limbs. And that can be very difficult to assess because we do not have, again, developmental information or embryonic information, and we only have so many fossils, and they're usually flat, and it's difficult to identify them. So again, MicroCT to the rescue. So in this particular study, we were able to actually scan small juveniles of this Lincolnia animal. And the one kind of doing the video on the left side is a 7mm long juvenile larva. Now, what you will see is, again, it has wonderfully preserved morphology. You can see every single limb, every single tiny spine on the limbs. It is just fantastic. And again, because this is a virtual model, we can actually modify and remove certain parts if we want to take a closer look to some part of the anatomy. And what we have is that we can see the mouth in, again, completely unprecedented detail for this kind of fossil. What we have here is that Lincolnia is showing that there is a mouth opening, which is what you see in panel B with an arrow, which is basically just a hole. But then associated with the mouth, we have this structure called the labrum. Now, the labrum is a flap-like feature in the mouth of all arthropods nowadays. And whether this was present or not in Lincolnia was actually extremely debated. Finding it allows us to actually make some correlations with the mouth of other kinds of arthropods. So for example, in panel D, you can see the mouth region of a spider, of a developing spider, of an embryo. And in panel E, you can see that of a cricket. So based on this information, we were able to tell that this animal, the Lincolnia, does have a labrum, and the labrum is quite reduced compared to what we see in the cricket. So this is supporting the interpretation that Lincolnia has a labrum, which is quite reduced. It helps us to clarify where these great appendages come from, which would be from the second segment of the head. And it provides additional support for the interpretation that this animal is closer related to chelicerate. It is not a chelicerate, it is not a spider at all, but it is closer to that particular evolutionary line than to any other line, as far as we can tell. And again, this kind of comes together quite nicely with data from morphology and from the nervous system in some other specimens. So this is basically my last slide. But what I hope to have achieved in this talk is to give you a sense of how much we do not know and how much these amazing fossils from the Cambrian can tell us, and just simply how amazing they are, because they truly are fantastic. This, what I'm showing right now, is an ongoing work by our local illustrator, Holly Sullivan, that is working with us in the MCC to produce some of these wonderful reconstructions to bring these animals to life, similar to the one I have behind me. And this is basically a Middle Cambrian locality, a Middle Cambrian environment from the Marine Formation in the House Range of Utah. It is one of those deposits that have started to get more attention lately, particularly, not exclusively, but particularly because of the research we do in our group. And again, we're able to reconstruct yet another one of these fascinating deposits in so much greater detail than ever possible before. So this is always a work in progress, and there is just so much more to come and so much work to be done. So hopefully we will have a lot of Cambrian for many, many years still ahead of us. With all that being said, I just want to, thank you, all of you, for kind of joining me today in this National Fossil Day, to thank all the wonderful people that make up the lab for invertebrate paleontology at Harvard, and all of which contribute in their own way to this research and to basically our collective experience and expertise. So with all of that being said, I am very happy to answer any questions, and thank you so much again for your time.
Javier, thank you so much for a wonderful presentation. Now, first, let me ask if you can close your presentation. I don't know if that's possible. Ah, very good. So let's see, we have several actually, what I'd like to do is not pay attention to the questions so I could just play with the Sketchfab graphics that I've downloaded onto my iPad, but I'll take a few minutes away from that to moderate the questions. So we have several outstanding questions submitted by several people. Let's kind of set this up like a TV game show. We have some questions which are real softballs. They should be very easy for you to answer, and then they go in degree of difficulty. So let's start with first from George. He asks, was Anomalocaris a predator or filter feeder?
Anomalocaris would fall within the predator category. That being said, there are many species of Anomalocaris, and some of them have been misinterpreted when they were first described. So for example, there is one called Anomalocaris brixi, which even though it is currently classified as Anomalocaris, it will very likely change classification because it is a filter feeder. But the correct Anomalocaris is definitely on the predatory side.
Okay, let's try another, hopefully a short one. We've got a lot of questions to answer here. So what kind of animal was Wiwaxia?
Wiwaxia is currently regarded as being very likely a close relative of either mollusks or annelids, or the group that joins both of them together, which are collectively known as spiralians. Wiwaxia had a muscular foot, a little bit like a snail. And it also has this kind of really intense armor of little platelets, which are, they are not shelly. They are more like just hardened, but not shelly. And they do have the mouth parts that look very similar to the mouth parts of a snail. So if anyone has ever bought, for example, these little aquatic snails that you can get and have as a pets, and they go and stick on the basically on the plastic, and you can see how they're scraping. Something very similar is going on with Wiwaxia. So it is definitely on that part of the tree, but it is also a particularly difficult animal to work with because it's so squashed and it has so much morphology. It can be very difficult to tease it apart and have a clear look at what's going on.
Okay, thank you. Now here's a very interesting general question, which I suspect a lot of people are interested in the answer. This comes from Michael. Wonderful Life was published 31 years ago. In view of all the more recent developments, is it still a valuable book to read? And if not, what book do you recommend for non-paleontologists?
Mm-hmm. That's a great question. I still read Wonderful Life because I think it's a wonderful, let's increase the vocabulary here. It is a great time capsule of how evolutionary thinking was during the 90s. And indeed, I use it as part of a course I teach in Harvard because it helps to make the point of how our understanding of these animals has changed and matured, and how context is so important for understanding this kind of data. Because paleontology has a peculiarity in that it has a very strong historical component to it. So understanding what people said before and why can be as useful as making new analysis. So just for that, I think it's still very worthwhile reading, and it is just a fun thing to read too. In terms of other books that you can consult for a more up-to-date perspective, there is a book called The Cambrian Explosion by Lou Gerwin and Jim Ballantyne. I think it was published either last year or 2 years ago. And that one does provide a much more modern view of what these animals are and their diversity and how we interpret them.
Okay, thank you. Now, let's see. Let's do this one here. This is your 100-point question. Spiders do not have biramous limbs, but early in development, they do have a second axis on their embryonic legs called gnathendites. Are crustaceans epipodites homologous to chelicerate gnathendites?
Wow, that is a great question. So actually, when I showed the figure, that one was showing one of these gnathendites. Now, the difference is that spiders early in their evolutionary history, so they're related to horseshoe crabs, which do have these spines, which would be homologous to the gnathendite. The difference is that all of these projections originate in different parts of the limb. The epipodites, so again, if you had my arm, so the epipodites come out on the dorsal, on the back, on the dorsal side like this, because they have to be external for water contact. The endites, which are spines usually, well, not usually, always on the ventral side, have a feeding function. So they have to be closer to the midline because the mouth faces backwards. So they have to produce this medial food group. So they will actually be on this orientation because they are used for chomping down on food. So what you see on the spider is to some extent a vestigial feature or hangover from this earlier ancestry. Because spiders nowadays have shifted all of their feeding functions to their fangs, their chelicera on the front. But if you see arachnid or spider relatives from the Paleozoic, some of them do still have some of these spines on their more proximal parts of the limb. So they're not homologous to epipodites, but they are rather a vestigial part of their feeding apparatus that is not present anymore in adult extant representatives.
Okay, thank you. Now here's a good one. This is a more speculative. You get the 100 points. The more speculative general question from Abel: what caused the Cambrian explosion?
I could sit here for 10 hours and still not have a definitive answer. And so it is a fantastic question, but it's also incredibly complicated to answer because the more we research into it, the more confounding factors there are. Some people suggest that there were environmental triggers that caused the Cambrian explosion. Like, for example, there is a correlation of increased oxygen in the environment close to the Cambrian explosion. There is evidence of strong tectonics happening before that would have input a lot of nutrients in the ocean waters. That could have caused a Cambrian explosion. There are events known as snowball Earth, which are global glaciations just before the Cambrian explosion. Heck, there are even supernovas and polar wonder happening before the Cambrian explosion. So it is very difficult to say, oh, clearly any of those alone is the result. Other people argue that the Cambrian explosion was basically a result of the increasing ecological complexity of the organisms that were already there that just happened to reach a critical mass, and then boom, everything just went sort of like super accelerated in terms of competition. Before the Cambrian, in the Ediacaran, we do have evidence for macroscopic life. But yet, I mean, the precise reason why we see animals at the Cambrian is still a little bit elusive. It has also been suggested that it may be that the Cambrian was very conducive to preservation because of the changes in the seawater chemistry. And what we see at the Cambrian explosion is more as an explosion of fossils rather than an explosion of animals themselves. But again, the true answer probably lies in a combination of all of these different factors. So again, there is no one golden ticket for the Cambrian explosion. It's more of a multitude of events coming together to produce this period in the history of life.
Great. Okay, here's actually a question that follows up the idea of preservation from Craig. Is there a chance of finding an unsquashed version of any of these critters someday?
I suppose it depends how much are you happy to settle for. At present, what I have shown is as close as we can possibly get of being able to take these fossils out virtually from the rock, as unsquashed as they can be, considering that they're still reasonably flattened. But these are macrofossils, so these are large fossils that we can see with our own eyes. During the Cambrian, there are other modes of preservation that do fossilize very, very tiny organisms in 3D, like in complete 3D. And this is through a process called phosphatization. So they basically become kind of replicated by phosphate, by sulfur-reducing bacteria. And in those cases, we have amazingly 3D preservation, but they're tiny. So you need to have an electron microscope to see them in any meaningful detail. And they also have a number of issues. They provide amazing information, but we do not know if they are the adults or the juveniles, or they're just so immature that we cannot really tell that much about some aspects of their phylogeny. So with exceptional preservation, it's always a trade-off. Either we have something awesome and we suffer on some other way. So it is very useful for that reason to investigate the fossil record from as many different perspectives as possible, because we can put all of this together and say something much more complete than the sum of it, well, than the parts alone.
Great. Here's just a technical question. Someone is asking you to repeat the name of the book by Doug Emlin and James Valentine.
So the book is called The Cambrian Explosion.
Cambrian Explosion?
Yes. My understanding is that there will be a follow-up email with for all attendees of this lecture, so we can include the link to that book in that email as well for all of those that are interested.
Great. Okay, let's see. We've got some more. You game to do a few more questions, Javier?
Oh, absolutely, yes.
Okay. What has been done recently in the past year in terms of research on the Qingyang biota?
The new one, like not Shenyang, the Qingyang.
Qing,
yes.
Jiang.
Mm-hmm. So it's difficult to tell because of the current situation we live in. I would imagine that my colleagues have not been able to go to the field as they normally would. So I would imagine that fieldwork has stopped significantly for the time being. However, there are tons of material existing at the moment in Northwest University in Xi'an. And I know that there are some of these projects ongoing. I am involved in a few of them. But again, things have slowed a little bit because of the disruptions around. But I can definitely assure that there are a lot of fossils, and Qingyang is very peculiar because it does preserve animals that we don't usually see even in exceptional deposits like comb jellies or jellyfish. So truly soft and snotty, and yet we have them, well, I mean, our colleagues have them in pretty healthy numbers in Qingyang. So it is going to really become one of the deposits to attract more attention in the following years to come.
Great. Okay, here are two, I think, related questions. One from Fabrizio. How do you think Cambrian feeding habits evolved? Did the carnivorous habit come first, then suspension feeders?
I guess it depends on what organism we are talking about. If we're talking chronologically, let's go chicken and egg. I would say I don't say. It's a fact that suspension feeding came first because sponges are extremely powerful suspension feeders in the oceans nowadays. They are animals. They do not have kind of complex bells and whistles, but they basically, all they do is sit and filter feed or suspension feed nonstop all the time, all day, every single day, to the extent that they're basically a major ocean pump. So definitely suspension feeding came first in animals. It depends when we look at specific groups of animals, whether predation is more of an ancestral feature or suspension feeding is more of a derived feature. I imagine this may be more geared towards the radiodons. And I would argue that for radiodons, I think that maybe predation may have come out first, because we have some relatives of radiodons that are a little bit different, but again, similar enough. And they do have more of the grabby, stabby frontal appendages than the suspension feeding ones. But again, it will be very dependent on the group of organisms we're talking about.
Okay, thank you. This next question I think also follows on to that, is related. It's very general, but it applies to what you just said. How are convergent traits distinguished from inherited from a common ancestor?
So that is another excellent question, and it is difficult. The more data we have, the better and informed hypothesis we can make to establish this distinction. The problem is that we are constantly struggling with this point in the fossil record, simply because we only have morphology to go with, right? So if we see two things that look very similar, well, is this similarity a result of convergence or a result of inheritance? In the case of Cambrian that I talked about, we have this very distinctive horseshoe-shaped shape, horseshoe-shaped head shield. Now, we can be pretty confident that that is a result of convergent evolution, because we have other parts of the animal in the Bruges shell specimens that show that this is definitely a radiodont. It has the frontal appendages, it has the circular mouth, it has the body flaps, and it is not a horseshoe crab, which has legs and has gills and has other parts and other bits and pieces. So the more data we put into the equation, the easier it is for us to say whether something is convergent or not. In other cases, when we have uncertainties, or simply we do not have enough information to be able to make this distinction, it is very difficult to tell, or even impossible, because both hypotheses may be as equally valid. Okay? So to give another example, one may argue, or Javier, I'm going to ask you you actually stop there, because I want to get another 2 questions in before we have to shut down for the night. As you say, you could go on for hours about some of these things. So just 2 more questions, and I want to apologize for those people who submitted questions, but we weren't able to answer through the talk. So the first one from Mark: instead of looking forward from the Cambrian, have you also looked back and attempted to make evolutionary connections to any Ediacaran fauna? Great question. I have not done this myself, because I am quite specialized and have a lot of work still to do with the arthropods and the relatives. But other colleagues have actually been much more involved in doing this. For example, a colleague called Jennifer Hoyle-Cuthill has been able to identify links between Cambrian fossils in Xinjiang and some of the Ediacaran fronts around the world. So it is being done, just not by myself at present.
Okay, this will be the last question. Again, thank you again, Javier, for a wonderful presentation, and thank you, audience, for some great questions. From Jaden: seeing all this diversity so early in Earth's history makes me think, how fast can nature fill in a niche? It's cool seeing just in one branch of life filter feeders, predators, and bottom feeders all at once. Do you think these niches were filled earlier and Anomalocaris and others outcompeted previous animals, or did the Cambrian explosion initiate most of it?
So just for a bit of context there, actually the Cambrian explosion comes in really late in the history of the Earth. For most of the Earth's life, most of the Earth's history, life is unicellular and mostly prokaryotic. There is even this period of time called the boring billion, because again, we have tiny things and not a lot goes on in terms of fossils. So the Cambrian explosion is actually a much later event, which means that actually having complex ecologies is something really difficult to achieve, and it takes a lot of time and a lot of evolution to accumulate to be able to interact with the environment in this particular way. In the case of the radiodons or the Anomalocaris, actually what happens is that, so some of the work by people in the lab, like Stephen Bates, is suggesting that actually Anomalocarids, the predators, were outcompeted by the filter feeders based on the distribution of fossils we see in the Upper Cambrian and even in the Ordovician. But again, this is more interesting research to come out that will clarify this situation.
Well, you did a wonderful job, Javier, both in your presentation and in fielding all of these questions. So thanks very much to you for giving this talk. We look forward to hearing a sequel sometime in the future. And thank you all again at home for joining us this evening. Have a good night. Bye-bye.
Thank you so much.